Hi, Lance.
Hi, Andre.
Lance is Associate Director of the CMM, Distinguished Research Professor, GRA Distinguished Investigator in Complex Carbohydrate, Research Center, University of Georgia.
All right, thanks, Andre. All right, so thank you everyone for joining us today. Uh, my name, like Andre mentioned, my name is Lance Wells. I've been working on OGT and OGLICNAC for 25 years. So I did my postdoctoral work with a guy named Jerry Hart, who's the guy who discovered the OGLICNAC modification, and have been working on it in my own lab since 2003. And so, Andre and I talked a little bit, and I've talked to some of the other families. And one of the things we wanted to do today is just kind of provide a little bit of basic information. Uh, right, everybody's kind of at different levels when it comes to science, right? So I'm going to try to keep it pretty basic. Hopefully, it doesn't confuse anyone. If it does, at the end, please just ask me questions. Uh, One of the things I need to start off with always is a disclaimer, right? I'm not a medical doctor. I have a PhD, right? I don't provide clinical advice, and you need to contact your physician for medical advice, of course. All right. And so, with that, let me just get started a little bit with trying to understand what we're all going to talk about, right? Because we're going to be talking about the OGT gene and genes and proteins. And so every cell in our body has a nucleus. Inside that nucleus are these things called chromosomes. And those chromosomes are made of DNA. And in that DNA, certain segments of DNA are what we call genes. And genes encode proteins. And so in humans, we've got about 24,000 genes that encode for proteins. And when you kind of think about proteins, right, proteins are what do most of the work inside our cells. They're kind of the engines, if you will, right? They do things like break down food into simple compounds, repair damage when it occurs. They're what it's involved in fighting infection when we get sick, muscle contraction, our heartbeat. They allow our neurons to transmit messages, right? So they're really doing all the work. Are these 24,000 proteins that humans have? And so when we talk about a gene, we're talking about a small section on a chromosome that encodes the instructions for making a specific protein. And as I mentioned, right, we're talking about 24,000 proteins. Unfortunately, right, mutations can occur in genes. And what that does is it generates an incorrect instructions for how to build this specific protein. Uh, and in some cases, that mutation, which will cause a protein not to be assembled correctly, will not oftentimes, but can lead to a human disease, right? And that's kind of what we're here to talk about, right? And so, uh, to back up, because OGT that we're going to be talking about is kind of an unusual gene because of what chromosome it's on, is all humans have 46 chromosomes, right? And so for 22 of those chromosomes, you actually have two copies, right? You have a copy from your mom and a copy from your dad. And these are what we call autosomal chromosomes. And the nice thing there is you have two copies of every single gene in your body, and for those 22 chromosomes. And so if one is bad, oftentimes you will not have disease because the other one is okay, right? Right, because you have two copies. However, right, if we go to the sex chromosomes, the X and Y chromosomes, if you're male, you have one X chromosome and one Y chromosome. So if you have a mutation on your X chromosome, you don't have another X chromosome that can keep you from having disease. Of course, also sometimes in females, just having one bad copy is enough to show a disease. And so when we talk about the X chromosome, we're talking about 1,000 proteins that are encoded with genes, and one of those is OGT, right? But the big thing to remember, and the reason why we see with any X-linked disease, which the OGT-CDG definitely is we see many more males affected than females, right? Because if you're male, you only have one copy of X, right? So you don't have this two copy issue. And so I'm going to show you this really complicated figure, right? Sorry, right? So this is the very first mutation that was ever described by Charles Schwartz in my laboratory back in 2015, so almost 10 years ago now, where this one female, so males are squares and females are circles, right on the screen, and if they have X-linked intellectual disability, there'll be a dark square, right? So there's 3 affected males, and this one female had 3 husbands and had, wow, 1, 2, 3, 4, 5, 8 children, um, one of which was affected. But then also the daughters also passed it on, and so this is what we call a classical X-linked inheritance, where we don't see affected females, we see affected males primarily, and that's kind of classical X-linked ID, and that's what we started with, was this initial mutation L254F. But between 2015 and now, the game has really changed, right? And so on the right here, I'm just showing you kind of a cartoon of this OGT protein, right? It's kind of got 2 domains, kind of an at the left side, a tetratricopeptide domain, and at the right side is kind of the enzyme function, kind of the catalytic, kind of the working horse domain. And you can see that there's mutations all over the place. And actually, this is incomplete, right? Maybe some of your family mutations are on here, and maybe they're not, right? Because it's incomplete. But we're already well over 50 families now with variants across the OGT protein that are displaying X-linked intellectual disability, which has now been referred to as OGT-CDG. When we first described it, we described it as OGT-XLID, but it's exact same syndrome. And so just a little bit about the O-glycnac transferase protein that's encoded by the OGT gene. This is kind of a picture of it on the right. Kind of if you could zoom in, right? It's obviously it's not something you can see with your naked eye, but if you could zoom in, that's kind of what the protein looks like. And so it's a pretty big protein, and it's localized inside your cell in the nucleus and cytoplasm. And so in terms of CDGs, this is what really makes this protein very different, right? So most congenital disorders of glycosylation, which there are a lot of CDGs out there, are for proteins that are either on the plasma membrane or being secreted from cells. And that's usually what we think about when we think about this, glycosylation, congenital disorders of glycosylation. But it turns out that there's one type of glycosylation done by the O-glycnac transferase protein that's inside the cell. So it's in your nucleus and your cytoplasm, the mitochondria, so inside the cell. And so this makes this very different than all the rest of the CDGs. The enzyme actually happens to dimerize, which is important for some mutations. Some mutations might impact that ability. It has this big repeating N-terminus, which is what's at the bottom of the structure here. And it has a lot of proteins that associate with it. And those associations are important for it to function correctly. And we have some preliminary data that many of the mutations in the TPR domain are actually changing who OGT can interact with. And that's probably changing what proteins get modified. And that's going to have some downstream effects. And of course, OGT is on the X chromosome, and it was at a region that had already been linked to neurological diseases, 20 or 30 years ago, before we actually knew about the X-linked intellectual disability. And of course, mutations right now are found in patients with X-linked intellectual disability. Like I said, more than 50 families. Right, I think we're probably approaching 100 patients. So what is this O-glycnac transferase? What's it doing? And so O-glycnac transferase is the protein that adds this sugar moiety onto other proteins, right? And so it adds an N-acetylglucosamine, or you'll often hear it called glycnac, right? O-glycnac. And so the O-glycnac transferase was actually originally cloned in 1998 in Jerry Hart's lab and John Hanover's lab. And then that's the enzyme that puts it on. There's also an enzyme that takes it off. And that enzyme is called O-glycnacase. And that was cloned in 2001 by Yuan Gao and myself while we were postdocs in Jerry Hart's lab. And so what we've been trying to study with this modification is what happens, right? So if we have a mutation in the gene causing a variant in the protein, are the steady state OGT levels down? And sometimes, right, in our cell lines, right, we have various cell lines. So sometimes the OGT levels are down, but not all the time. Sometimes OGA levels are altered, right? There's no mutation in OGA, but it's trying to compensate for a change in OGT activity. And so OGT levels are altered sometimes. And then finally, O-GlcNAc, this modification that occurs on all these hundreds and hundreds of proteins in your cells, right? Those O-GlcNAc levels get altered sometimes, right? And so one of the things we're really struggling with is we haven't found something that happens all the time in an X-linked intellectual disability patients with OGT. And so that's one of the things we're really working for, right? So those are called biomarkers, something that happens always if you have this disease. And so that's one of the things, my lab and many other labs are looking for. One of the other things to think about, right? So we're adding this sugar, but where does that sugar come from? And that sugar originally just starts off as glucose, right? Which we all consume vast amounts of, probably in America more than we should usually. And that glucose can get modified a bunch of different ways to form this UDP-GlcNAc. And that's GlcNAc is that UDP-GlcNAc is what's used by OGT to put O-GlcNAc onto other proteins. And then, of course, OGA can take it off. And so one of the thoughts is people have been thinking about, right, in cell models, animal models, is substrate replacement therapy, right? Is can we feed more substrate, either that being glucosamine or N-acetylglucosamine, right? And would that possibly improve O-GlcNAc levels and outcomes, right? And we have no idea, right? We don't know the answer to that. Those are early day studies, but it's something that people are definitely considering. So I'm not going to go through this. This is I'll make these slides available. This is just what does O-GlcNAc do, right? So O-GlcNAc gets added to certain proteins, but so what, right? So what is it doing? And the reality is it's doing a lot of things. It changes that chromatin we were talking about, those chromosomes. It can change their structure. It can change what genes get turned into message, what messages get turned into proteins, what proteins interact with one another, what proteins get secreted from the cell, right? So it can do a lot of things. And in general, people tend to think about O-GlcNAc as a glucose sensor and regulator of energy homeostasis. So it's a way to know how much energy the cell has, and that affects the cell's response to different stimuli. And that's kind of the overarching theme. The problem is we have thousands of proteins that get modified by O-GlcNAc, and so are there key ones that are responsible for what we observe in the X-linked intellectual disability syndrome? And that's one of those $1 million questions we would really love to know the answer to. So having said that, let me just back up a little bit and talk about some of the current research, right? So right now, what many groups are doing, right, including ours, is trying to elucidate the impact of the variants, right? So you saw that there's different mutations all over the gene, and so we're trying to take each one individually and say what's going on, right? And also to search for common biomarkers if we take them all together. And so to do that, we can actually purify the enzymes out of cells and study their activity and their stability, and we do that. We can actually generate cell lines that carry those mutations and ask how do those cells behave. And then more recently, we can develop animal models, right? So that could be Drosophila, right? Fruit flies. That could be mice, which Dan von Alten's group has got some mice studies that have been published now. Or it could be rat models, which is where my laboratory is interested, or even higher models, right? It could be, primate models eventually. But I don't know anyone who's doing that right now. And so that's kind of what we're doing as basic scientists, right? But the other things we're doing is examining the impact of intervention at all levels, right? So in cell lines, on purified enzymes, in animal models, what happens if we adjust substrate levels? If we feed cells glucosamine, if we feed them glycnac, right? If we feed them just more glucose, right? So looking at substrate therapy outputs. Uh, the other idea is to elevate glycnac levels potentially with OGA inhibitors. So if you inhibit the enzyme that removes the modification, then levels will build up of the glycnac. And OGA inhibitors have been developed for some other purposes, primarily things like Alzheimer's disease and Parkinson's disease. There's some thought that OGA inhibitors might be useful there. And so there's some companies that have actually developed some really nice OGA inhibitors and OGA inhibitors that have already gone through phase 1 clinical trials, which are safety trials in humans successfully. And so OGA inhibitors are attractive as a possibility. And then, of course, long term, right, there's gene therapy, right? I don't know if some of y'all maybe have heard about CRISPR-Cas technology. It won the Nobel Prize a couple of years ago. It's received a lot of press as a way to repair mutations in the genome. And we can do that in cell lines. We can do that in animal models, right? But doing that in humans, right? That's a very, very long-term goal, but something, right, that obviously many people with congenital diseases are very interested in studying. And so with that, I tried to keep it kind of short because I'd much rather have a dialogue than just me preaching at you. So here's a picture of my lab from a little while ago, all the people that actually do the work. And thank you for your time. I'd be happy to answer any questions. And again, I just want to make it clear that, right, I and my lab, we don't provide medical advice. If you need medical advice, please contact your physician.